individuals with Down syndrome. He's an expert in aging-related issues and cognitive phenotypes associated with trisomy 21. He's also involved in developing and testing the efficacy of treatments and interventions for adults with intellectual disabilities. And he leads the Neurodevelopmental Disorders Clinical Trials Center, which conducts phase 2 and 3 clinical trials for treatments of autism, Down syndrome, and other conditions. Professor Strydom is the current president of the Trisomy 21 Research Society. And we are looking forward to his talk today.
Thank you. Thank you for that introduction. I hope everybody can hear me. Yeah? I was just testing if you could hear me.
Yes, yes, sir, you are absolutely audible. Kindly share
Okay,
your
so
screen.
I'm going to share my talk with you.
Yes, it is absolutely visible and you are absolutely audible. Thank you, sir.
Great, thank you.
It's fine.
So, so thank you for that introduction, which means that I don't have to introduce myself other than to say that I'm from the Institute of Psychiatry, Psychology and Neurosciences, the old IOP at King's College London. We're based in South London, and so this is the view from South London across the Thames to the Building of Parliament. What I'm going to be focusing on today is Alzheimer's disease in people with Down syndrome. But first, just a brief reminder of what Alzheimer's disease is. Alzheimer's disease, the pathology of Alzheimer's disease concerns the deposition of amyloid in the form of plaques extracellularly, followed by the development of neurofibrillary tangles, which is, contains tau, another protein. Something which is not often appreciated is that amyloid can also be deposited along the vessel walls, causing the vessel walls to become weak and brittle and causing bleeds, microbleeds and strokes. And this is called cerebral amyloid angiopathy. And together, this neuropathological process leads to neurodegeneration or loss of cell material. And the typical atrophy that you can see in the top picture associated with Alzheimer's disease at post-mortem with enlarged ventricles. Alzheimer's pathology has been known to occur in people with Down syndrome for nearly 100 years now. The earliest report of Alzheimer's disease in a Down syndrome person that we could find, Alzheimer's pathology in a Down syndrome person that we could find, is this paper from 1929 in a German journal describing a new staining method for plaques, and then also a case series of individuals in whom they've observed plaques, and they mentioned that the youngest person in which they have seen plaques at after death was a person with Down syndrome. Of course, it was called something different in those days. A person with Down syndrome, who I think was around 40 years of age, and so. Later on, it became much more apparent in later neuropathological studies that Alzheimer's disease is a common feature in the brains of people with Down syndrome after at post-mortem. And so this is a recent paper by Professor Mann and his group in Manchester. And on the top line, you can see ages of different individuals with Down syndrome. So it starts age 0 to 13, and it ends at age, I think, 76. And it shows the development of Alzheimer's pathology starting around the age of 35. So you can see around the age of 35. I wonder if I can have a highlighter. I'll show around here. Starts amyloid pathology, and by age 50, virtually everybody with Down syndrome have significant deposition of amyloid in their brains. And that's followed by tau pathology. So again, that starts a little bit later, maybe in the late 40s. And then after 50, it's a very prominent feature at death. And one can also see CAA, or cerebral amyloid angiopathy, developing. At clinical level, we now know that the lifetime risk for Alzheimer's disease in people with Down syndrome is approximately 90% by age 65. By that we mean, and this is based on work done by Mary McCarran in Ireland. She followed a group of individuals with Down syndrome for more than 20 years to estimate this risk. So the onset of clinical symptoms may occur in the vast majority of people with Down syndrome if they all lived to the age of 65. Of course, people may die still at younger ages due to other concerns. And in the UK, we have pooled a number of dementia cases with Down syndrome from various clinics across England, and estimated that the mean age of diagnosis of dementia in Down syndrome is around 55, which is 2 to 3 decades earlier compared to other people with Alzheimer's disease, with an interquartile range between 50 and 60, meaning that 25% of people may develop symptoms or be diagnosed with dementia before age 50 and 25% after age 60, but the majority between age 50 and 60. We can also track the development of dementia symptoms. So in this case, we were using data from our longitudinal study, the Lundowns cohort, and we excluded people who already had a diagnosis of dementia and just looked at how many people develop symptoms over a period of 2 years. And you can see cross-sectionally, we established that if you exclude people with an established diagnosis, 1 in 5 of individuals aged 35 to 45 may have memory problems. So this is the graph we're looking at. And between 46 to 55 in red, 1 in 3 may have memory problems, but not yet diagnosed with dementia. And over the age of 56, it will be 2/3. And we estimated the incidence, new onset of dementia symptoms, to be 25 per 100 person year. So 1 in 4 of people over the age of 35 will develop some degree of dementia symptoms over a period of a year's follow-up. Moreover, dementia and Alzheimer's disease is associated with high rates of mortality. So in our longitudinal study of individuals with over the age of 35, we identified that 70% who died during a 2-year period of follow-up had a clinical diagnosis of dementia before they died. And most of the rest actually showed clear signs of dementia, but were not diagnosed with dementia yet, including seizures. New onset of seizures in individuals with Down syndrome over the age of 40 are almost invariably due to Alzheimer's disease. It's a common complication. And so we concluded that Alzheimer's disease is now the main cause of death in adults, not in children, but in adults with Down syndrome. It's the main health concern for adults with Down syndrome. It's an incredible burden of disease. And just some further data on survival. So typically in the general population, individuals diagnosed with sporadic Alzheimer's disease can expect a mean survival, years of survival for around 7 or 8 years. So we estimated that if an individual with Down syndrome is diagnosed with dementia before age 50, then their average or median survival is around 5 years. Between age 50 and 60, they can expect to live another 4 years. And after 60, the median survival is 2.5 years. Given that most cases of sporadic Alzheimer's disease in the general population is diagnosed after 60, and people have a survival rate of around 8 years, it's also the case that individuals with Down syndrome, once they have developed Alzheimer's disease, have a much shorter survival rate. So very briefly, just touching on genetics. I'm not a geneticist, so I'll keep it brief. The genetics of Alzheimer's disease in people with Down syndrome. So Down syndrome is, of course, due to having 3 copies of chromosome 21, with 3 copies instead of 2 of chromosome 21 genes. And some of these genes are dosage sensitive, including amyloid precursor protein gene on chromosome 21. And all Down syndrome individuals with full trisomy 21 will have 3 copies of this gene, which is associated now with Alzheimer's disease. And we know that it is the most important genetic factor associated with Alzheimer's disease in people with Down syndrome, because rare individuals with Down syndrome without an extra APP gene, maybe because they have a partial trisomy 21 or a translocation, which doesn't involve the APP gene, it's very rare, do not seem to develop Alzheimer's disease. So these individuals seem to be free of the disease. And then conversely, rare individuals with another condition called duplication APP, in this case, these people have a sort of a copy number variant, which means that they have 3 copies of this part of chromosome 21 gene, only this part, which contains the amyloid precursor protein gene. They don't have the critical region of Down syndrome. That doesn't include the critical region of Down syndrome, which is probably around there. In other words, they don't present with the features of Down syndrome. They don't have intellectual disability, but they all develop Alzheimer's disease at similar age as people with Down syndrome. It's a very rare condition. There's only about 20 or 30 families with this condition identified in the world, but nevertheless, genetically extremely important to show the effect of amyloid overdose, amyloid protein overdose. And so this amyloid protein overdose was one of the key discoveries to explain the amyloid cascade, what happens if there's abnormalities in amyloid production. And I'm showing a picture of Professor John Hardy, who's one of the geneticists who in the 1990s described the amyloid cascade, and he's also a member of the Lundowns Consortium. So what happens in people with Down syndrome is that they have overproduction of amyloid and the breakdown product amyloid beta, which then eventually aggregates and accumulates and leads to toxicity with cell death. And it's after cell death that we can diagnose dementia. In people who do not have Down syndrome, in the general population, the issue is probably to do with aging. So they have normal amyloid levels, normal amyloid beta levels for most of their life. But with aging, clearance becomes an issue, clearance of amyloid from the brain. And again, it leads to aggregation and accumulation with toxicity and cell death. The problem at the moment in terms of treatment is all the treatment trials seem to target this time when people are already symptomatic or diagnosed with dementia. But unfortunately, this is quite late in the process then. It is already when people are experiencing cell death. And really to make a difference, at least from an amyloid kind of target, we would need to start treatment much earlier when amyloid first starts to be deposited in the brain. We cannot do that in sporadic Alzheimer's disease, by the way, because we can't predict who will be developing sporadic Alzheimer's disease. It's always difficult to predict, but we can in people with Down syndrome, so keep that in mind. We can also track the in vivo deposition of amyloid now using amyloid PET scans, and this is a graph, a very famous graph now from our colleagues in Cambridge, Anthony Holland and Sheed Zayman's group. They used amyloid PET scans, and they've shown that you can demonstrate people becoming amyloid PET positive between the age of 40 and 50 if they have Down syndrome. So everybody converts from being amyloid negative to becoming amyloid positive by age 50. These scans pick up a slightly later form of deposition than we can see on neuropathological studies, and that's why there's a slight age difference. We can also measure amyloid beta, one of the breakdown products of amyloid protein, in the periphery, in plasma. using single molecule immunoassays, a very sensitive way of measuring protein or protein components. And doing that, we have shown that individuals with Down syndrome have much higher levels than controls. So much higher mean levels of amyloid in their blood than controls, higher even than those with sporadic Alzheimer's disease. Although I should say that this biomarker is not a very good biomarker to use clinically, because amyloid may also be produced peripherally. And the other issue is that rather than increasing as people develop dementia, amyloid peripherally actually decreases. So that's why sporadic Alzheimer's disease cases here have lower amyloid beta 42 levels, slightly lower than controls. The idea is that it gets deposited in the brain, and so it reduces in the periphery. So pooling data from other studies in a meta-analysis, we have shown that quite clearly amyloid beta 42, which is one subtype of amyloid beta, amyloid breakdown, is definitely increased in people with Down syndrome, as is amyloid beta 40, two different lengths of amyloid. The significance of this is that amyloid beta 42 tends to be deposited in plaques, whereas amyloid beta 40 tends to be deposited along the vessel walls. I'm going to skip that slide. So within the Lundowns Consortium, we were interested in also trying to see how the risk for Alzheimer's disease in people with Down syndrome might relate to development, early development. The idea being that, you know, something that sort of is bad for your brain and an older age might also be bad for you during development, or perhaps not. And so we collaborated with our colleagues in Birkbeck, where they recruited a number of infants with Down syndrome, infants and small children with Down syndrome, and did some simple computerized tasks, eye tracking tasks. So the kids are shown a screen, and on the screen they can see a little figure which is animated. So and they wait until the child is engaging with that, is looking at the figure. And then there's a gap, for example, the screen goes blank. And then later on, when the screen comes on again, there's another figure on another part of the screen. And they measure how long the child takes to disengage from the first to the second position. And you can do that simultaneous and overlap design as well. So using that approach, we then looked at ApoE, apolipoprotein E, another gene and protein that's been identified as a risk factor for sporadic Alzheimer's disease due to its effect probably on vascular factors. And we surprisingly observed that babies with ApoE4 carriers did better with engaging faster, disengaging faster their attention than those who did not have ApoE4. While so here you can see another graph showing that after a while, though, by the age of 5, ApoE4 and non-E4 carriers perform similarly on the task. There's also a developmental effect. So children get typically better on this task as they get older. So there's the age. So what happens in adulthood, though, in older adults, we see the reverse effect. So again, it's an attention type task that we're reporting here. The ApoE4 cases do worse. They take much longer to complete the task than those who do not have ApoE4. So there's kind of like antagonistic pleiotropy of ApoE. So it has a sort of a positive developmental effect for ApoE4, but then a negative effect with aging. From a genetics point of view, this is quite interesting and might explain why the ApoE gene has been retained, or the bad allele has been retained evolutionary. So moving on from the genetics then, thinking a little bit about the symptoms now, the development of Alzheimer's disease in people with Down syndrome. So that's kind of difficult to establish in some ways because we know that people with Down syndrome have unusual features in their brain. So typically they have hyperfrontality, so smaller frontal lobes, reduced hippocampus, even before the onset of dementia, as well as cerebellar hyperplasia, but relative sparing of the trunk and posterior cortical structures. They also have typically smaller brains than their peers. So this relates to a cognitive profile, a typical cognitive profile, which carers will tell you if you ask them that individuals with Down syndrome typically have better non-verbal abilities than verbal abilities, so can do things better than they can explain what they're doing. And within verbal abilities, understanding receptive language is better than expressive language. So expressive language is a particular issue. They may have a relative weakness in short-term memory versus longer-term memory. So carers may say, oh, he can't tell you what he did this morning, but he can talk in detail about a party he attended 2 months ago, something like that. And visuospatial short-term memory is better than verbal memory. So this is important to remember in education, for example, that verbal teaching methods is not so good for people with Down syndrome. They need to do something more visuospatially in order to remember certain things and to be trained to do tasks. So I'm not going to go through all the differences in much detail. You can look at it in the on the screen, but one other thing I wanted to highlight is because of the smaller frontal lobes, there's also a relative weakness in prefrontal systems, including executive functioning, so planning, attention, and so on. And does this mean that people with Down syndrome present differently when they present with dementia? To some extent, yes, but largely it's the same pattern. So you have a deterioration in complex cognition, first of all, and then a decline in memory function before people develop more functional issues. What is unusual in Down syndrome is that you have a decline in executive function relatively early on, possibly because of the smaller frontal lobes at the start with behavioral and emotional changes alongside memory problems. Typically in sporadic Alzheimer's disease, that would happen later. And also prominent neurological symptoms later on, which again in sporadic Alzheimer's disease would be a very late sign, but here in people with Down syndrome, it may occur slightly earlier: incontinence, mobility issues, and in particular seizures. So this may mean that dementia diagnosis could be less reliable in people with Down syndrome. However, we have established by talking to clinicians and testing clinicians' dementia diagnostic abilities that if they're used to working with people with learning disabilities and they adapt by focusing on change from an individual baseline, from the person's own individual baseline, and make sure that they exclude other causes of decline. This is very important because people with Down syndrome can sometimes develop other issues that could look like dementia but isn't and is treatable. And this includes things like thyroid problems, hypothyroidism early throughout life. It's important to exclude that. Visual problems, including cataracts, hearing issues. Those are all things that need to be checked and excluded before we make a diagnosis of dementia. But if we do that, then clinicians are fairly reliable in terms of their diagnosis. So if one clinician says somebody has dementia, then another one tends to agree. And also the different diagnostic systems perform similarly, although there are some differences. So what about tracking change, cognitive change as people get older? So what we have done is to think carefully about assessment. So just showing our sort of typical battery. We also do MRI scans now in our longitudinal study. But in terms of cognitive test issues, we had to carefully think what we would use because we cannot use the usual kind of tests, including many mental state examinations, because of the threshold being high and involving writing and reading, for example, that many people with Down syndrome might not be able to engage with. So there's typically floor effects. And the other issue is that people present with a range of cognitive abilities. So you can have people who are very able, and then others who will struggle with certain tasks. So what we had to do was to use both individual tests and carer ratings on the same domains and adapt scoring levels and difficulty levels where necessary in order to make some of these cognitive tests suitable for people with Down syndrome. You can read about that in some of our papers. We've published it. And doing that, we've shown that we can track change as people get older. In this case, we're simply comparing older adults with younger individuals at peak of performance, and showing the significant drop-off in cognitive performance in older adults, starting probably with more subtle changes in memory or attention, so memory and attention after age of 40, and then rapidly progressing so that by age 55, most people have significantly poorer performance compared to younger peers. And we can specifically focus on memory, which we know memory decline is strongly associated with Alzheimer's disease. Here we're using a test called the Calcutta computerized test. So people see a screen and we tell them that in the hidden, and you have to remember where the present is. In this case, it's the pattern, the yellow pattern, and the screen goes white again, and after a delay, they have to touch the screen where the pattern was hidden. And so using this kind of approach, we can see a significant decline in memory function as people get older, long before the typical diagnosis of Alzheimer's disease at age 55. So you see a steep decline as people age. And we can also pick up differences between those who do have dementia or are symptomatic and don't have dementia. So using our battery of tests, we then applied some statistical modeling approaches, in this case, something called event-based modeling, which estimates an ordering of binary events from data from normal to abnormal, as it were, so that we can sort of establish a sequence of change, sort of a machine learning type of approach. And using that kind of approach, we've shown that the early stages of decline here is defined by memory and sustained attention tasks. And then after that, you get sort of general cognitive and executive functioning changes. And at the end, late stages is defined by informant ratings of function. And this is usually what triggers the dementia diagnosis. So there's sort of like a staging effect, a clear staging effect. And using the staging effect, we can also show the ApoE4 effect, a non-chromosome 21 gene. So at the same age, say age 50, a person with Down syndrome, age 50 with ApoE4 allele would be at a later stage of decline than one with an ApoE2 or 3 allele. So I want to next move on to thinking about what the next steps are in terms of research. What we really need to be focusing on now is thinking about how we can prevent this onset of pathology and clinical decline in people with Down syndrome. And we need to be getting ready for prevention trials of Alzheimer's disease and Down syndrome. And this is tricky because you need to start treatment early, before significant pathology has developed and well before clinical onset, and then treat for quite a while before you can see an effect. So that's quite a difficult situation in which the whole Alzheimer's disease field is struggling with. So what we want to do, for example, in memory decline, is to shift the typical memory decline that we see in people with Down syndrome, the very steep decline, to a much more subtle decline, which might be more like a typical normal aging. And so we need to power trials for showing this effect. And we also need to identify the age range at which we need to target for this kind of work, for a kind of prevention trial. And so here we are working on other statistical modeling methods, in this case using modeling from pharmacological studies, dose effect studies, to see if we can identify the sweet spot for a clinical trial where you could start to give a treatment and then expect to show that people do not drop off the cliff, as it were, as expected. So in the case of the Cantab-PAL, you would have to start around here to show that you're not getting this kind of effect in the treatment group. We could also then, once we've identified the age ranges to target, we can then compare different cooperative tests to see which one would be an optimal test battery in terms of sample sizes to use. And there's just some examples of what we might select. Another way of speeding up clinical trials and improving clinical trials in people with Down syndrome would be to use biomarkers. Here is one that we're quite excited about at the moment called neurofilament light. It's a neuronal structural protein associated with cell death, in other words, with neurodegeneration, which can be measured in plasma using Simoa assays. And using this method, we have shown a steep increase in neurofilament light levels as people with Down syndrome age. And we can also pick up differences between those with dementia and without dementia. And so this could be a way to see whether a treatment has an effect even before you're able to show a clinical effect. So if you can shift this change on a biomarker, then you know you are in the right, you've got the right treatment and you need to take it forward in larger trials. And the work was, our initial work was then replicated by Juan Fortea's group in Barcelona, where they replicated neurofilament light levels both in plasma as well as in CSF. Interestingly, we can show that neurofilament light levels in people with MCI, so early type of symptoms, are similar or comparable to people with confirmed dementia. So it shows that we can perhaps pick up early changes with this biomarker compared to those who do not have dementia. Oops, sorry, I'm going backwards now. And just very briefly, this is work that's been submitted recently. It's comparing people with Down syndrome on the basis of neurofilament light levels against other neurodegenerative conditions or potentially neurodegenerative conditions. Of course, neurofilament light is not specific to Alzheimer's disease. It may pick up other forms of neurodegeneration as well. But the interesting thing is that in Down syndrome, because Alzheimer's disease is the cause for neurodegeneration in the vast majority of people, we can quite clearly show the difference between individuals with Down syndrome without and with Alzheimer's disease. And that levels are typically higher in people with Down syndrome, particularly those with Alzheimer's disease, than other forms of Alzheimer's disease and most other neurodegenerative conditions. So to end, I would like to just talk a little bit about some of the things that we can do at the moment, the kind of care that we can provide to people, given that there isn't a treatment that can cure Alzheimer's disease. But there are still many other things we could do to improve the lives of people with Down syndrome who have been diagnosed with dementia. So I wanted to highlight again, firstly, the need to exclude comorbid conditions or conditions that can mimic Alzheimer's disease. I've mentioned hypothyroidism, I've mentioned visual and hearing problems, but sleep apnea is another thing that's increasingly become apparent. It's estimated that more than 50% of individuals with Down syndrome may have significant sleep apnea, so breathing issues as they fall asleep, with potential oxygen supply issues to the brain. And obviously this could affect cognitive functioning, and it's important to screen for this in people with Down syndrome. It's difficult to screen for it though, because ideally you need to do a PSG, a polysomnography, and people don't often engage with that. But we're working with our sleep lab to use home monitoring systems where people wear something called a watch pad, so a watch that measures pulse and oxygenation and movement, and could perhaps be a good way of establishing whether somebody has sleep apnea or not. And we've also been working on dementia pathways. So firstly, an assessment pathway to say how we will assess dementia, and a post-diagnostic dementia pathway. So what happens after people have been diagnosed with dementia? And we realized that there are a few things that we're not doing very well. One was breaking the news. So, you know, it is an upsetting thing to diagnose somebody with dementia. And so we had to work with psychology colleagues to think carefully about how we break the news to the service user, to their friends, and to their families. And we've also had to introduce advanced care planning. So that's the idea where at the very earliest stage, you go, you start a conversation with people about what will happen next. How do they want their care to be delivered to them as they progress in the condition? And we offer what we call bespoke training to staff and carers working with people with dementia. So our team would go into the care home or the family home, and then offer them specific training about how to manage a person with dementia and what they can expect. One of the key things is that people realize that it's a progressive condition, and they know what to expect, and also know how to manage problems when they develop, such as mobility issues. I'm not going to go through this, except to mention we use a tool called QOMIT to monitor quality of care, so we can see when care starts to become insufficient. And later on in the course of problems, we also need to think about end-of-life care issues. As you will have seen earlier, typically survival is 4 to 5 years, even less in older people. And so it's important to think about what end-of-life care will look like, and put together an advanced care plan involving palliative care specialists, ensuring that we would manage discomfort as soon as possible, and thinking about where the person would go if they can't manage in their own home. I want to finish on just mentioning acetylcholine esterase medication. So we do use this. We prescribe this to most people with Down syndrome who are diagnosed with dementia. It's a symptomatic treatment, so it's not going to make a change to the pathological process. But we do think that early on, there are some benefits, particularly for acetylcholine choline esterase inhibitors rather than memantine, for example. So we can see some effects on mortality, for example, which may be due to various factors, because this is not trial data, but nevertheless, people seem to tolerate the medication relatively well, and you do seem to have some sort of symptomatic benefit in the early stages of disease. So in summary and in conclusion, we can measure cognitive change before onset of dementia in people with Down syndrome, working on better outcome measures, though, in anticipation of clinical trials. There are now promising biomarkers associated with the development of Alzheimer's disease, not just neurofilament light, but potentially also new tau biomarkers. What we still need to understand in Down syndrome is how these relate to onset of symptoms over time, how it predicts the onset of symptoms over time. And we need to be thinking about doing clinical trials based on rational drug targets. So there are some drug targets, some new drugs available, some of which are targeting the amyloid pathway, others that may be targeting other aspects, tau and so on, or oxidative stress. And I think the key will be to think carefully about what age ranges of people to include in such trials and what outcome measures to use, depending on the stage of disease that you're targeting. And we can do a lot of things to maintain quality of life through good diagnostic support. So we can already help people to have a good quality of life despite having Alzheimer's disease. And so all that remains for me to do is to thank all our collaborators and colleagues that have been working together with us on the Lundowns Consortium, particularly my team, the people who have been helping to assess hundreds of people with Down syndrome, and the people who have helped us to recruit people with Down syndrome across England. So a large number of clinicians have been helping with the recruitment efforts. Thank you. And our funders, finally. Thank you.
Well, thank you, Professor Stridham, for your excellent and informative presentation. I think participants receive a lot of new informations regarding Alzheimer's disease in Down syndrome. And we have many parents from Down syndrome families. So the parents, I think, receives a lot of new informations and idea regarding the management of the Alzheimer's disease and their care of their children. So now this session is open for interaction. We received so many different questions in Zoom platform as well as in YouTube streaming. So may I ask you some questions from the participants? The first question is, why Down syndrome patients are prone to developing AD in their 30s?
Well, so that's to do with the genetic risk. You will remember that we talked about APP triplication, so amyloid precursor protein, of which they have three copies rather than the usual two, and it results in overproduction of amyloid, which then eventually which sort of needs to be processed, but it overcomes the processing process, and so eventually leads to deposition. This is the idea, theoretically, in any case, deposition of amyloid in the brain, which is toxic to the brain and causes the pathological process. So the way we think about it nowadays is that Alzheimer's disease in people with Down syndrome is somewhat different from sporadic Alzheimer's disease in that it is a genetic condition. In people with Down syndrome, it's definitely a genetic condition. And in that sense, it's similar to the other genetic types of Alzheimer's disease, familial Alzheimer's disease, such as, all the amyloid, the APP mutations, and the presenilin mutations.
Well, the next question is, accumulating evidence indicating our trend to indicate a role of impaired insulin signaling and glucose metabolism in the progression of Alzheimer's disease. Do we see them happening in Down syndrome patients as well prior to developing AD?
Yeah, I mean, that's an interesting idea. I think that, the dementia process is associated with lots of changes. And there is, of course, the idea that insulin signaling, glucose metabolism, and so on could be associated with the more vascular, the more sort of vascular risk or the vascular pathology that's common to Alzheimer's disease, particularly in sporadic Alzheimer's disease. Now, we know that people with Down syndrome may be at increased risk for diabetes, for example. Although not many people are diagnosed with diabetes, there is ultimately some increase in risk. How that relates to Alzheimer's disease within people with Down syndrome, I don't know. I don't know of work, though, but that's an interesting question. It's something interesting to explore.
Yeah. The next question is, how soon we can detect AD biomarkers? How reliable they are in predicting the outcome and severity of the disease?
Yeah, that's a good question. So we can detect the biomarkers. As I mentioned, neurofilament light, for example, we can see an increase from age 40. Amyloid biomarkers, particularly amyloid beta in blood, we can see well before that, I think throughout the life of people with Down syndrome, they would have fairly high levels of amyloid beta in the blood, but in a way that just shows that they have too much amyloid which is being excreted. The question is how that relates to clinical onset of disease, and I think that's still a question in the broader Alzheimer's disease field that lots of people are working on. So there's all these new biomarkers, they're all very exciting, but what we need to establish, and unfortunately that will require some longitudinal work, is how a biomarker change relates to a change in clinical onset of disease. And we need to establish that in order to, before we can use it in clinic and before we can use it in clinical trials. So there's a lot of work going into it. So I should emphasize that these biomarkers don't have necessarily clinical relevance quite yet, but we're hoping that they would do in the next few years.
Well, and the next question is, does ApoE affect cholesterol level in Down syndrome?
Oh, that's a good question. Yes, we're hoping to look at that. So there may be a relationship between ApoE and cholesterol in general, and we're hoping to look at that in people with Down syndrome specifically at some stage. Okay. It's slightly difficult in the sense that ApoE4 alleles, I think it only occurs in 1 in 5 people, also only 1 in 5 people with Down syndrome, and so it's relatively rare. So getting enough people with different alleles is a little bit difficult.
Well, the next question is myotonic epilepsy associated with Alzheimer's disease in Down syndrome?
Yes, so typically what we see, so the seizure development as dementia progresses is quite common in Alzheimer's disease in people with Down syndrome. So we think we're in our longitudinal studies, we identify about 73/4 of people with dementia eventually develops seizures. So it is something that is a common presentation as the disease progresses. It's not often an early presentation, but it's often when the disease becomes a bit worse. And so what happens typically is a sort of myoclonic jerking. So we typically first see the kind of it's a sort of involuntary jerk as people wake up. So just a sort of a startle kind of response in the mornings or throughout the day, sudden startle. And then eventually that may progress then to seizures of, more generalized seizures. And so it does have a sort of myoclonic kind of picture. However, it is quite treatable, so it does respond to treatment with anti-epileptics. So we tend to get advice from neurologists very early on when people have experienced seizures, or even when they start to show the myoclonic jerks, and consider using some anti-epileptics, such as Keppra, for example, and it works quite well.
Well, the next question is, there are different proteins that are upregulated or downregulated in cerebrospinal fluid and blood during Alzheimer's disease, but still now no protein expression-based diagnostic model are in market or haven't been developed. Can you please give an idea which are the causes of this failure?
Yeah, I mean, like I mentioned earlier, it is true that it's early days and these biomarkers can't quite yet be used as in the clinics. But I'm not a biomarker guy, so I'm not a chemist, but the biomarker people are really optimistic that we will have reliable biomarkers in the very near future, but there's some more work to be done. It might be a combination of biomarkers, for example, denoting different stages of disease, for example. I'm particularly interested in peripheral biomarkers. So in people with Down syndrome, I think it's very difficult to do lumbar punctures and get CSF, but it will be much easier to take a blood sample. So our own lab collaborate with the biomarker people to think about peripheral biomarkers, blood biomarkers.
Well, the next question is, do you think the traditional IQ and cognitive assessment are suitable for Down syndrome people?
Sorry, say that again?
Yeah. Do you think the traditional IQ and cognitive assessment are suitable for Down syndrome people?
Some tests can be used in people with Down syndrome. So we do IQ tests, but we use a brief IQ test usually. So we use the KBIT-2. So it's a very brief kind of IQ test. The problem with IQ tests is that when you use standardized scores, then most people are at floor, so score below IQ 40. However, longitudinally, we find that you could use the raw scores of IQ tests to track change over time. It doesn't tell you the person's IQ, but it helps to track change over time. There are some other IQ tests like LITER 3 that might also be suitable, and some colleagues use those. But again, you would have a floor effect. So, more than half of people, or around half of people, will score at the floor. So we'll have an IQ of 40 and below. Cognitive assessments, other cognitive assessments typically need to be tweaked a little bit. They need to be made easier, and you need to think about floor effects. So, for example, the Cantab-PAL test is one developed for the general population, a memory test, visuospatial memory test. But we had to make sure that we included lots of levels at the start of the test, the easier stages of the test, in order to pick up subtle differences between people. If you started at too complex level, then people just score at the floor. They just do poorly on the test, and you can't pick up differences in people who are struggling a little bit with the test. So that's just an example. Clinically, there are other tests that can be used, like the CAMCOG-DS. So that's a test that we use in our clinic, more sort of comprehensive cognitive assessment. And you may look into that as a possibility in clinics. And so it's sort of a composite test.
Well, almost two related questions. The question is, several social and environmental interventions seems to be effective in supporting people with dementia. Does the same non-drug treatments be effective in DS?
Yes, yeah, that's an interesting idea. So we're thinking about things like, well, it depends on whether you're thinking in terms of prevention. So there's the idea that healthy lifestyles, exercise, and so on could delay some degree of decline. And I think it should also be true in people with Down syndrome. There hasn't been many trials, though, testing that, and it's something worth thinking about it. And then, of course, there may be other social and environmental interventions that would be applicable to people with Down syndrome and dementia. So I'm thinking of things like certain forms of cognitive stimulation, keeping people active and busy. We always advise them to continue to engage as much as possible in their usual activities, to continue to go to day centers and so on, because it's good to keep the person interested and engaged. It also helps with their orientation. And then, of course, there are lots of environmental changes that could be put in place to ensure safety of the environment and to help the person orient to their space. Pictures on doors, for example, colored tracks on the floor so that they know the yellow track goes to the kitchen or something like that. And because of the visuospatial issues that develop with dementia, we also often think about the bathroom, how we can make the contrast between the toilet and the floor greater so that it's easier for people to use the toilet. So these are all interventions that are important.
Well, the next question is, Can microdose lithium treatment of bipolar disorder, stabilize cognitive impairment in patients with Alzheimer's disease?
Oh, I don't know. There has been the idea that lithium could be neuroprotective, could have neuroprotective effects. And I know there was a pilot trial of lithium in people with Down syndrome to see whether it kind of reduces the onset of Alzheimer's disease. That trial is not published yet. It was too small, I think, to show an effect. And I think there are some people who are still interested on some of the lithium pathway, as it were. So there's certainly options there, and it's an interesting idea that I think will remain live for a while, because there are theoretical reasons why lithium might have, or similar drugs might have potential effects.
Well, another interesting question, is there any influence of sex on the manifestation of AD in DS patients?
So just before I go on to that, just to say, there's no evidence for microdose lithium use in people with Down syndrome. So I wouldn't recommend that as a treatment yet, but it's certainly something worth considering in terms of clinical trials, just to be clear about that. In terms of sex differences, I mean, overall, we think that the risk is similar in people, in men and women with Down syndrome. So the overall risk for the developing dementia is very similar. So different from the general population, where you do see prominent sex differences. There may be subtle differences in terms of age of onset of disease, you know, the age at which people develop symptoms. And so we still need to do some more work to understand those a bit more clearly. There has also been some earlier work that showed some relationship to the onset of the menopause. The idea being that if menopause is shifted earlier in those people that's been shifted earlier, you also get earlier onset of Alzheimer's disease. Again, maybe thinking about some of the vascular issues and endocrine related changes or pathways that might impact on Alzheimer's pathology. So again, there needs to be more work in those areas.
Well, and some question from YouTube also. One interesting question. Does tau protein deposits more in case of Down syndrome than other Alzheimer's disease?
Does tau protein do what? Sorry?
Does tau protein deposit more in case of Down syndrome with Alzheimer's disease than only Alzheimer's disease patient, non-syndromic Alzheimer's disease patient?
Yes, it looks like it's similar. It looks like it's fairly similar. I can't say it's more or less. Certainly seems to be at least the same, as in there may be some subtle neuropathological differences, but in the main, we think that sporadic Alzheimer's disease pathology is very comparable to Down syndrome Alzheimer's disease pathology.
Okay, with this all, we are just closing this question answer session. Thank you, Professor Andre Stridham, for your kind consent to be with us. We are really grateful to you for sparing some time for us from your very tight academic and administrative schedule. For participant, once again, I just want to mention Professor Andre Stridham, presently in chair and president of International Trisomy Research Society. And he is very busy in different academic and administrative work. In spite of that, just on our request, he consented to deliver this wonderful and very informative and enlightening lectures for our participant. Thank you, Dr. Stridham, and see you again. We will going to meet again within a couple of hours in our COVID task force meeting. So that's all about, and goodbye. And I hope in near future, we will invite you to India in our any conference organized by T21RS Indian chapter. Thank you, Andre.
Thank you. And thank you for the invitation. It's a great program. Thank you.
Thank you. Bye.
Am I audible? Am I audible, Dr. Ghosh? I think our next invited speaker, Mara, has still now not joined. So may I ask you to continue the session with Dr. De and Pinku? Participants, please bear with us for just a few minutes because the next panelist or next invited speaker is yet to join.
Well, so we are waiting for our next panelist, and before she join with us, we are just going for the short talk by a research scholar from our team at Calcutta. So may I request Professor Papiya Ghosh to introduce our research scholar to the participants. Professor Ghosh, over to Professor Ghosh.
Thank you, Dr. Ghosh. Now, there will be a short talk on the topic polymorphism of maternal genes for folate metabolism as risk factor for Down syndrome birth. The talk will be delivered by Pinku Haldar, who is the senior scholar of cytogenetics and genomics lab, and he is basically an UGC fellow. His area of research interest is maternal folate metabolism, gene mutation, and Down syndrome baby birth risk. So I do request Pinku to share your screen and start your presentation. It's over to Pinku.
Hello. Am I audible to you, ma'am? Hello.
Yes, you are absolutely. Yes, Pinku, you are absolutely audible and your screen is visible. Please carry on. Thank you.
Okay. Thank you, ma'am. Good evening, respected delegates and friends. I am Pinku Haldar from University of Calcutta, India. And today, the topic of my presentation is polymorphisms of maternal gene for folate metabolism as risk factor for Down syndrome birth. So let's start. Polymorphisms of maternal folate regulator and their association with Down syndrome birth risk was initially identified by James et al. and then evaluated in different ethnic population across the world. But unfortunately, Indian Bengali population remain unexplored. So the objective of our study was to find out whether there is any association between maternal folate pathway gene polymorphism and Down syndrome birth risk in Indian West Bengal population as a function of maternal age and type of meiotic error. But before going into the details, we should know about what is folate and why folate is so important. Folate is a component of vitamin B complex, also known as vitamin B9. It is the natural form which is easily available in green leafy vegetables, whereas folic acid, also known as terryl monoglutamate, is the synthetic form of naturally occurring folate, and the efficacy of this folic acid is much more higher than that of the naturally occurring folate, because this monogglutamated form can easily cross the cell membrane and enter into the cell. And that's why this folic acid is extensively used in different nutritional supplements and fortified food products. Here is a simplified diagram of folate metabolic pathway. And in our study, we have focused only on these 4 genes. RFC1, also known as reduced folate carrier 1. Another name of the gene is SLC19A1. It is actually a transporter. It helps to transport folate from blood into the cell. Next is MTHFR, that is methylene tetrahydrofolate reductase. It is a central enzyme of folate metabolic pathway. It catalyzes the conversion of 5,10-methylene tetrahydrofolate to 5-methyl tetrahydrofolate. And this 5-methyl tetrahydrofolate, actually the physiological active form of folate, it can now enter into 1-carbon metabolism and donate its methyl group. Next is MTR, that is methionine synthase. It remethylates homocysteine into methionine. And for the activation of this methionine synthase required another enzyme, that is MTRR, or methionine synthase reductase. It is a vitamin B12 dependent enzyme. The methionine so formed then converted to SAM, that is sulfur adenosyl methionine. And SAM act as intracellular methyl group donor in various reactions of DNA, RNA, neurotransmitter, etc. There are different types of methyl transferases which transfer methyl group from SAM, and then SAM is converted to SA, that is sulfur adenosyl homocysteine. And in the next step, SA is converted to homocysteine by the action of enzyme SA hydrolases. So this folate metabolic pathway is very important for maintenance proper DNA methylation pattern. Here is a pictorial representation of the location of all 5 polymorphic variants that we have studied. As you can see, MTHFR and MTRR gene is located in chromosome number 1, whereas MTRR gene is located in chromosome number 5, and RFC1 gene is located in chromosome number 21. Next, materials and methodology in brief. It was a hospital-based case control study. We collected our blood sample from Kolkata and its surrounding hospital. Blood samples and epidemiological data were collected from 1,019 Down syndrome trios, and we termed them as our case sample, and also from 730 age-matched control trios who have normal euploid baby and no previous symptoms of abnormal pregnancy loss or chromosomal disorder, and we termed them as our control sample. Blood samples were collected only after taking full consent from the participating families under the guidance of our physician friends. And our study was supported by the Ethical Clearance Committee of University of Calcutta as well as IPGM-ER Kolkata. So in step 1, we did karyotyping, serum folate and homocysteine levels were measured, and genomic DNA was isolated. In step 2, We genotyped all the families with panel of chromosome 21 specific STR marker for characterization of parental origin as well as detection of maternal origin of meiotic error. In step 3, polymorphisms of folate metabolic pathway were detected by bidirectional Sanger sequencing method, and then we did suitable statistical analysis for interpretation of our result. And now the result part. Here is a graphical representation of maternal serum folate and homocysteine level in case and control group. As you can see from the bar graph, serum folate level in case group is relatively lower than that of the control group, whereas serum homocysteine level is relatively high in case group than that of the control group. Out 1019 case sample, 922 sample shows maternal error, whereas 97 sample exhibit paternal error. And we excluded this 97 sample from our study because we are looking for maternal risk factor. And out of 922 maternal error, 785 sample shows maternal meiosis 1 error, whereas 234 sample shows maternal meiosis 2 error. Sorry. And polymorphism analyzed for all 922 samples, and here is a bar graph which represent frequency of all 5 polymorphic variant that we have studied in M1 and M2 error group. And as you can see from the bar graph, the frequency of all 5 polymorphic variant is very, very high in case of M2 error group, whereas it is negligible in case of M1 error group. So we considered M2 error group as our case sample for further analysis with age-match control. Next, minor allele frequency of variant in case and control. Here is a bar graph which represent changes in minor allele frequency in case and control. Out of 5 polymorphisms studied, MTR-A2756G, MTRR-A66G, MTHFR-A1298C, and RFC1A80G shows significant difference in minor allele frequency in case and control, and it is statistically very, very highly significant. Whereas in case of MTHFR-C677T polymorphism, no significant difference in minor allele frequency is observed. Next, association between maternal risk genotype and Down syndrome birth risk. For MTR-A2756G polymorphism, we take homozygous dominant A genotype as our reference. AG heterozygote was associated with approximately 3-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically highly significant. Whereas homozygous mutant GG genotype doesn't exhibit significant association in case of our West Bengal population, and A to G substitution in one or both allele associated with approximately 2.9-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically highly significant. Next, for MTRR-A66G polymorphism, again in this case we take homozygous dominant genotype as our reference. AG was associated with approximately 2.48-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very highly significant. Homozygous mutant GG genotype doesn't exhibit any significant association in case of our West Bengal population, whereas A to G substitution in one or both allele associated with approximately 2.54-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very highly significant. Next, for MTHFR-C677T polymorphism, none of the genotypic combination exhibit association with Down syndrome birth risk in case of our Indian Bengali population. Next, for MTHFR-A1298C polymorphism, again in this case we take homozygous dominant A genotype as our reference, and AC heterozygote was associated with approximately 2.21-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very highly significant. Whereas homozygous mutant CC genotype doesn't exhibit any significant association, and A to C substitution in one or both allele associated with approximately 2.2-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very highly significant. Last, for RFC1A80G polymorphism, we take homozygous dominant A genotype as our reference. AG heterozygote was associated with approximately 6.75-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very, very highly significant. For homozygous mutant GG genotype, the p-value that we have got is 0.151, which is not statistically significant, and A to G substitution in one or both allele associated with approximately 6.41-fold increased risk of Down syndrome birth, and the p-value that we have got is statistically very, very highly significant. Next, we also detected probable damaging effect of all 4 polymorphic variants that appear significant for our Indian Bengali population in transcript level using different in silico software program. Here is a pictorial representation which exhibit result of transcription factor binding in wild type and mutated sequence, and here the red box indicating changes in transcription factor binding due to polymorphism. At last, we also predict functional prediction of polymorphic allele through in silico analysis, and different in silico softwares were used to predict the polymorphism status, and almost all in silico software program predict this 4 polymorphism as deleterious or probably damaging. And now the discussion part. When there is no polymorphism or metabolic block in folate regulator, proper DNA methylation level is maintained, and it ultimately results in proper segregation of chromosome during cell division, resulting formation of healthy gamete. On the other hand, when there is polymorphism or metabolic block in folate regulator, it in turn results in low level of methionine production, and this low level of methionine in turn results in low level of sulfur adenosyl methionine production. And as I earlier mentioned that SAM involved in various intracellular reaction, so low level of SAM doesn't ensure proper maintenance of DNA methylation level, which ultimately result in DNA hypomethylation, and this DNA hypomethylation may result in improper segregation of chromosome during cell division, resulting aneuploidy like trisomy 21. So in a nutshell, our study revealed that folate regulator polymorphism are risk factor for maternal meiosis II non-disjunction in oocyte in age-independent manner. In case of our West Bengal population, the polymorphism MTR8275G, MTRRA66G, MTHFR A1298C, and RFC1A80G exhibit strong association with Down syndrome birth risk, while the polymorphism MTHFR C6770 is not a risk factor for our West Bengal population. However, synergistic study revealed that when this risk allele is present in association with other risk allele, it increases their risk level. First of all, I would like to thank all the participating families for donating their tissue sample for our research work. I would like to thank Department of Zoology, University of Calcutta, Department of Science and Technology, Government of India for funding in our project. I would like to thank University Grant Commission, Government of India, and Trisomy 21 Research Society, Netherlands. I would like to thank my guide, Dr. Sujoy Ghosh, for allowing me to present in this webinar. I would like to thank Dr. Sumantro Sarkar. He is our physician collaborator. I would like to thank Professor Stephanie Sherman and Professor Eleanor Fingold. They are our overseas collaborator, and this is Cytogenetics and Genomic Research Unit. I would like to thank all my lab mates for always helping me and motivating me in my research work. But before ending, I just want to say 2 to 3 words. Believe me, they can do everything like a normal person. We just need to love them a little extra, care them a little extra, and support them a little extra. So don't judge them, just love them. Thanks to all of you.
Thank you, Pinku. You have few questions that I think you can address after our next invited speaker will complete her session.
Okay, sure, ma'am. Sure, ma'am. Thank you.
Thank you very much.
Welcome, ma'am, to the T21RS Indian chapter.
Yeah, welcome, Monica. So this is the time to hear from Professor Monica Lakhan-Paul. May I request Professor Subroto De to switch on his camera and come to the webinar. Professor De. May I request Professor Subroto De to introduce Professor Monica Lakhan-Paul. Hello, Professor De. Hello. Hello.
Sir, please unmute yourself and
Sir, please unmute your speaker and start camera. Professor De, Professor Subroto De, please. Unmute করো এখান থেকে। এদিকে অন করো। Ask to unmute করো। Ask to start video করো। Professor De. Sir, okay. It's the time to hear from Professor Monica Lakhan-Paul is a professor from University London, University College London. He's and Professor De. Oh, Professor De is on here. Professor De, the Vice Chancellor of Vivekananda University, India, and he is my PhD supervisor, and he is pioneer in Down syndrome research in India. May I request Professor De to introduce Professor Monica Lakhan-Paul to the participant. Over to Professor De. Professor De, can you hear me? Professor De, can you hear me? Please introduce Professor Monica Lakhan-Paul.
I think sir has some network problem.
I think so. Professor De. Hello, sir.
I think sir has some network issue.
I am introducing Professor Monica Lokhonpal. Professor Monica Lokhonpal is a professor of integrated community child health at University College London. She is deputy theme lead for the collaboration in leadership in the applied health research and care North theme, adjunct professor at Public Health Foundation India, and also provost UCL South Asia. Professor Lokhonpal graduated with a Bachelor of Medicine from University of Manchester in 1992 and with a Doctor of Medicine from University of Nottingham in May 2003. Lokhonpal also gained her doctorate in the pediatrics and the child health in 2003. Asian Women of Achievement, she won Asian Women and Achievement Award in the 2010. Professor Lokhonpal has received many prestigious awards that include HSJ Patient Safety Award for spotting the sick child in the year 2011, selected as NICE Fellow for 2010 to 13, awards for founding clinical director of the National Collaborating Center of Women and the Child Health. Her work spanned from clinical trial to the participatory methods, including public engagement and working with the arts and humanities to improve outcome for vulnerable children, with a focus on the UK and India. She particularly focusing on disability, asthma, nutrition as an exemplar public health issue in the South Asian families to optimize their health and later risk for non-communicable disease. So it's a time to hear from Professor Lokhonpal. Professor Lokhonpal, please share your screen.
Thank you. Can you see it? It says that I'm sharing it. Can you see it?
Yeah.
I think Pinku has not stopped sharing.
It says I am.
Yeah. Sharing to bondho koro tomar.
That's why she is facing problem. She has given in the chat.
It says that I am sharing.
Yeah. Okay, so you can share.
I am in. It says that I'm sharing.
Yeah, but we are not.
Yes, this screen is of ma'ams, I think. You have presentation ml.doc and cheapbook.docs. This screen is probably of ma'ams.
Yeah, mine shows it's sharing.
Yeah, exactly. Oh, it's fine.
Yeah,
absolutely fine. Absolutely
fine. Perfect. Sorry about that. So, apologies to everybody for the technology and the timing and everything else. A big apology. What I wanted to spend my time today is really opening a conversation rather than doing a formal presentation. I will do a formal presentation, but I want to give this an opportunity to really engage with people's questions, and if they have questions, to ask me. Because I think you've heard a lot of science, and that's what you're here to hear, but there's also, I understand on the webinar is parents, there's also people from other backgrounds as well. And so I think it's important that we can talk from all those contexts and understand what Down syndrome is about and trying to bring some of the science together. And what does it actually really mean for the children? So yes, I spend a lot of my time doing research. I could spend a lot of time showing lots of detailed graphs and data on some of the research we do. But actually what I want is that since you've been hearing about all the research so far, we just really stop and think about what this actually means for children. I have a real passion of translational research, applying the research into practice and actually making a difference. One of the things that I really feel passionate about is the fact that it's important for us to do the research, but if the research stays in a journal, if it stays on a shelf, and if we don't actually have to take the moral duty up to move that research into practice, we're not really helping the children that we're here to serve. So some of what I'll be talking about today is really the practicalities of how we translate our research into practice and what that means for the children. So as you can see from the title here is Improving the Quality of Life for Children with Down Syndrome, the importance of early recognition of comorbidities. What we know from Down syndrome is that many children will have comorbidities that range across from heart disease to eyes to many, many different factors that we need to consider. And that's what we'll be talking a little bit about today. Just to go back into the history, and you may have covered this, but just as a reminder for everybody, and I know you're all experts in this area, and I think it's important to remember where the terminology came from. So John Langdon Down, an English physician, was the first to publish an accurate description of a person with Down syndrome in his work in 1866. And unfortunately, at the time, the terminology that was used was called Mongolism. And this really wasn't very helpful to the children or the community. It made children feel as though they were separate, different in some sort of way than everybody else. So Down syndrome itself became an accepted term in the early 1970s. And it was a term that most families were acquainted with, most families would find accepting. But there are still many families who don't want their child to have a different name or a different label than all the other children who they play with or spend time with. In addition to that, if we look at this paragraph that I've extracted in 1860s, in John Langdon's paper itself, it uses the word congenital idiots, instances of degeneracy arriving from tuberculosis in the parents. So there in this short article, people are already making presumptions about what it means for a person who has Down syndrome, or indeed, where has Down syndrome come from? Whose fault is it? Those terminologies, you know, where did it originate from? That discussion was already happening, but as you can see, the terminology is not very positive. So I think in science, we actually have a duty of care of considering what is the terminology that we are using? What's the terminology that we are perpetuating? How do we engage with the community? How do we engage with the children and give them the best opportunities for the future? And thinking very carefully about how scientists use the language in communication for their own peers, but also with the families as well. Another reason for this is if we go back and think, historically, people with Down syndrome had very few rights, and they were treated quite poorly. From around 1850 until the 1960s, people had learning disabilities, many who were actually institutionalized as well. If we actually go back and have a look, we've seen that they had poor living conditions, lack of education. Many were not integrated into education at all. Deprivation of healthcare. They were not prioritized for surgeries. Some of them nutritionally were also starved as well. And many children, once they became adolescents, also had to endure forced sterilization. In the 1970s, the right to education, to comprehensive healthcare was brought in. And it was thought that children, all citizens should be equal, and they should all have good access to healthcare, including a range of different specialties. That's when the thinking about early intervention programs came in. How can we actually work with parents at an early stage in the child's trajectory life course to bring about changes and improved outcomes? How can we support good parenting skills and ordinary family life? And education and support groups were starting really to give some peer support. So this in turn led to opportunities for people with Down syndrome. They were entering school, they were able to leave home, able to set up new relationships, gain employment, and lead large independent lives, which probably wasn't there before. Article 23 here as well, child with a disability has the right to live a full and decent life with dignity and, as far as possible, independence, so that they can play an active part in community. Governments can do all they can to support disabled children and their families. And this is something that we were actually talking about with the last speaker. You know, how do we ensure that these children grow up into adults who can have a good, engaging life. What is it that as doctors and as scientists we should be doing to prepare the children for their future? And part of that is advocacy with the governments. Government policy is hugely important for change. And what we need to be doing is advocating for our young people to ensure that they have the housing, the education, the healthcare systems in place to be able to bring about this change. So when we talk about research, yes, we need the biological research, we need the science research, but we also need the social science research. Very, very important that we have people from all disciplines working together to consider what is the impact on young people and adults living in the community, and what can we actually do about that. So let's try together to ensure that no one suffers unnecessarily. If there are symptoms that can be treated, how do we do that? And we should ensure that everybody reaches the potential that they can without medical problems. So as I said, we're all making advancements in science, and advancements in science ensure that we can actually improve outcomes as well. However, advancements in science sometimes are helpful, sometimes not helpful. So we know that there's been a current dilemma of early prenatal diagnosis of Down syndrome, and some of that work was done in our own university. The positives were that mothers could identify if they had a risk of Down syndrome at a very early age of the baby, of the conception. The negative side is that actually it made mothers and women, or it could make mothers and women, this is the debate that's been going on, is does that put women under pressure to have to make a choice about delivery of their child? And now I'm not going to answer that question, but this is a question of science. On one hand, science is great. It tells us more than we knew before. On the other hand, there are ethical and moral discussions that have to happen around those scientific findings that we have. This is the medical advances that started. It feels like I'm giving you a bit of a history lesson, but actually this is where it all started. And it was, again, in Jerome in 1959, the French physician identified Down syndrome as the chromosomal disorder. And that really changed a lot. And the reason I'm bringing that as a reminder to everybody was, if you remember what I put as the slide earlier, when it talked about parents having TB, was this caused by an infection? Then in 1959, we started realizing that actually it was a chromosomal condition. Now, again, one would think that that scientific advancement was a wonderful breakthrough. We knew why this was happening. And if you know that it's a genetic disorder, you can actually, could you do something about it? The question is, why should we, and do we want to? Again, another big ethical debate. One of the issues that have come up with this is that once people start considering a disorder as a genetic disorder, then again, from the social science perspective, you start seeing social cultural influences on the discussions that start having. Whose fault is it? Whose chromosomes are there? Which gender does this come from? Does it come from the mother? Does it come from the father? Even though it may not be linked to science at all, the question is often blame of parents and whose fault it is. So again, we need to have this integrated approach to research where we're also thinking about how we engage with the community, how we share information effectively, and how we support the families who have children with Down syndrome in a very positive way. The other finding we know is that children are now living longer as adults with Down syndrome. And I know that you've had previous talks on the long-term effects of Down syndrome on the brain, on Alzheimer's disease, and what that really means. And that again is extremely important, because if children are going to live into adulthood now, we really need to be making very, very clear steps to improving their outcomes and their well-being. So in 1910, children with Down syndrome were expected to survive to about age 9. Discovery of antibiotics, because as we know, there's risks of infection with Down syndrome. But with the discovery of antibiotics, the survival rate increased to about 19 or 20. And then there were more improvements and advancements made. Surgery was possible, corrective heart surgery was possible. So as many as 18% of adults with Down syndrome in the UK reach age 60, and many may even live a lot longer than that. Now, of course, globally, this will vary. It will vary according to many, many different aspects. It will vary where you are in the world. It will vary according to the nutritional access you have, what technology you can access, what medicines you will access. So I'm giving a picture of the increase of age of individuals, but that age will be impacted by many, many different factors wherever you live. So this is another graph as well that shows from some of the research as well, which shows, again, an increase in life expectancy. So the life expectancy, and this is from Australia, has, you can see, been increasing. So I'm come to the way I try and address most of my research, whether it's children with Down syndrome or whether it's children from with other complex problems as well. And I always use the basis of the socio-ecological model as a basis for many of my discussions. And you can see in this socio-ecological model, these concentric circles. Within those concentric circles, you can see that we talk about the individual, and there you can see genotype. What is it? What is the child's individual genotype? How can science help with how we consider that genotype and what impact that has on the child? But then, as I was alluding to earlier, what is the knowledge of the family? What are the beliefs of the family? What are the experiences of the family? And how does that family unit and that family network live within community, the social network they have? And within that, what about the organizational network, the school, different religious institutions? Who are the actors in that in a religious institution or in that community? And then what about the environment that the child lives in, the school, where they go to play, where they go to learn music, where their peers are? And then, of course, policies, laws. What is it that the law and the policies can do to actually support the human rights of these individual people? So what that comes to is that, you know, we cannot live in our silos. We can't do our research in our silos. We can't do our research in a lab and then presume that's going to have an impact on the children. We can't do research that's solely in one area of the research without integrating that with the other specialties as well. For example, if you're doing research related to health, what about the considerations of the environment that the child lives in? What about the considerations of the educational system that the child lives within? Now that's important because here I come to some facts. You know, 80 to 85% of brain growth takes place during the first 2 years of life. That's for any child. Of course, with Down syndrome, there are certain challenges that we have to overcome. But generally, in children in general, your brain is growing at the fastest rate during the first 2 years of life. That means for children with Down syndrome as well, what can we do to ensure that that time period of the child is really when we focus most of our attention? A good brain growth also is influenced by good nutrition, stimulation, whether this be play, positive parental interaction. The reason I'm bringing that up is because children with Down syndrome, nutrition is one of the key aspects that we should be focusing on. Sometimes a lot of time goes on, you know, their heart disease or their other infections, which is, of course, a big challenge to live with. But one fundamental need of any child is nutrition. And if we can consider what good nutrition means for a child with Down syndrome, are they getting their vitamins? Are they getting vitamin D? Are they getting iron? Are they not deficient in any of their nutrients? This will give a good start to the child. Talk about play. Again, we need to be doing more research on the impact of play on children as well. So nutrition and play often go together, development often goes together. And that interaction is important because if a child is, as we know, if a child is left by themselves in a corner and is not interacted with, their developmental growth will slow. The more we play with a child, the more we give and have interaction with a child, the more likely it is that that child will be able to progress. And that's with a child with Down syndrome or without Down syndrome. Positive interaction with the family and parents as well. The more we play with the child, the more we look at the child, the more the child can interact back with us. Again, it can be a challenge, and parents or families may feel they don't want to trouble the child, or the child's quiet, so they don't need the interaction. But actually teaching parents that you need this positive, continuous interaction is hugely important. And there is more and more research being done on educational attainment, educational resilience, how to interact with children. And I think that's a space that we should watch very, very carefully, is what is it that we can do to actually support the brain growth in the children. So we need an integrated approach, which I've said before, and giving the child the best at the start of life will improve their outcomes later on. But this starts before a child is born. It starts with the mother's health. And I think one thing I want to reiterate today is the later we leave actioning all our interventions on children, the worse the outcomes will be later on in life. But this starts antenatally. It starts during childbirth, it starts around childbirth, and also with the mother. So if a mother, whether they know their child has Down syndrome or not has Down syndrome, ensuring that that mother's health, the mother's nutrition is actually at a level that we would want it to be, with all the nutrients, all the vitamins, everything that a mother should have for childbirth, that's hugely, hugely important. So let's not start too late. So what I'm saying is let's take a life course approach. Let's think of the whole life course, because if we do, it'll be more likely that our children will have a good and positive life. So if we think of the newborn with Down syndrome, we're aware that that's where our intervention should start. Children are more likely to have congenital heart disease. They're more likely to have gastrointestinal problems, more likely to have cataracts, transient abnormal myelopiosis, which means in the bone marrow, the cells, the generation of the cells, prolonged jaundice, poor feeding, and that comes back to nutrition again, which is sometimes missed, and slow weight gain. So from our research perspective, what have we been doing? We've been doing some research where we've been looking at large databases and looking at what are the comorbidities. So this has been most of our research recently. What are the comorbidities that we should be watching out for? What are the key comorbidities that we need to be thinking of in children? And the reason for that is if we don't know what the comorbidities are, we're not going to target our energy, our time, our investment in the healthcare system to identify those key comorbidities. And children will be left, we'll be picking up the pieces later on, we'll be intervening at a late stage. And actually, if we proactively intervened, we could prevent some of the long-term problems. And Down syndrome specific growth charts, I presume everybody is using those, but it's interesting when you come out of the Down syndrome community, how many people don't know about the growth charts, how many people are not using the growth charts. And why that is important is if we use the growth charts as a way of identifying problems early, we will be able to intervene early. And like I said, this is something that's really international, the growth charts are, and we would hope that it was being disseminated everywhere, but it not necessarily so. And the problem with that is, if we put a child on a growth chart that's not a Down syndrome specific growth chart, their height and weight may be perfectly fine for themselves, but on another growth chart, it may not be. That means we may intervene when we don't need to. We may raise anxiety where we don't need to. And then that has knock-on effects. So again, one of my pleas and one of my discussion points that I repeat is that we often spend a lot of money on very expensive science, important as it is. Actually, we forget that we need to be researching some of the basic factors. You know, are people using growth charts? Do they know how to use them? Why are they not using them? Very, very basic implementation science research must not be forgotten. The other side is management. If we think of when a baby's born, how many places set up their teams as multidisciplinary teams from the outset? Often children are sent from one person to another and miss that early support system. how can we do health systems research where we bring together team members, pediatricians, therapists, audiologists, ENT surgeons, ophthalmologists, educational experts, cardiologists, health visitors, all of it, a holistic team to actually look at that child from the beginning to later on in their life. Many different services around the world are set up in different ways, and it's understandable why that happens, but actually it has been shown that if you have an integrated team around the child, sharing information, the children are more likely to get optimal care. So let's think of the comorbidities of the health issues in school children. Again, this is some of the data that we've been looking at is what are the comorbidities at different age groups. So we know vision is a problem. Children don't like to wear their glasses. So even if they have visual problems, they're not wearing the glasses. That's a huge problem. Hearing, if you can't hear and you can't see, how do you develop? How do you get educated? How do you respond to your parents? Another issue is that often hearing and vision are not prioritized. The child doesn't want to wear their hearing aids. The child doesn't want to wear their glasses. Yet the sensory system is hugely important for children with Down syndrome. And giving children that opportunity through hearing aids and in glasses is really, really important and mustn't be missed. There is an association we're finding with children with celiac disease, and some protocols have started suggesting that we should be actually checking children for celiac disease, again, to intervene early so that their weight doesn't fall. Obstructive sleep apnea, again, we know that children, you will have heard them, sometimes we ask, do they stop breathing? Are they sleeping properly? And there's been a lot of research showing that there is a connection between obstructive sleep apnea and children with Down syndrome. What we do in our surveillance now is ensure that we ask parents, do they know if their children have stopped breathing at night or make funny noises? Now, there is a debate about then, do you do a sleep study on every child? Do you do a sleep study on every child every year? When is the optimal time to do this? And this debate it will be different due to access of healthcare in different countries. But one thing that we do know is if you do intervene and increase the oxygen level, that's good for the heart and it's good for the brain. So again, what can we bring into a surveillance system that actually is going to be very, very important for these children? Problem is often cost. Can we afford everything that we would like to do? We know that there's risk of infections, and we are doing some other research looking at the risk of respiratory infections in children with Down syndrome. We know that children with Down syndrome do have an increased risk of respiratory infections and end up in hospital more frequently. Sometimes children do seek healthcare for that. Sometimes they seek it quite late. And again, how do we alert parents to the need to seek care when they do? Epilepsy, we talked about skin. Now, skin I've put on here, but often skin is something that's actually missed. Again, from our data that we have collected, it is evident there are skin disorders, sometimes we're not sure what they are. Is it eczema? Is it something else? Blisters, risk of infections on the skins, more commonly with children with Down syndrome. And yet this can cause poor quality of life because of the itching and the pain to the child. And yet it seems to be something that is lower on the priority. This is anecdotal, but lower on the priority of seeking care for. We'll come to mental health and behavior, congenital heart disease, autoimmune diseases. We know that diabetes is higher in children with Down syndrome, thyroid disorder, vitiligo, alopecia, numerous things, blood disorders, and what we call cervical spine instability, which means that your bones are just here in your neck, move forward slightly. And there is a risk of the cervical spine and the bones in the neck here just pressing on the spine. Very, very, very rare. But in our surveillance, we always ask parents to be vigilant about this. And if the child stops walking, can't use their hands properly, they must seek help immediately. And that's something that's often forgotten. It's something that I'm very, very active about reminding parents about. But often when I see other people, it's something that may be forgotten in the routine healthcare surveillance. Health issues in adolescence, very, very similar. But you'll see that growth is sometimes something that we talk about because children may be shorter. Obesity, because they may have had joint problems. We know that arthritis may also be more common. They may have obesity because they can't run around. And there may be some behavioral issues as well, where children are eating or they're given food because we want to see them happy. And food and happiness seem to be related in many, many families. And so the children put on weight. The problem is that if they put on weight, they get more joint problems, they get more heart problems, they can't engage with activity. And also on top of all of that, the future risk of diabetes will go up. We always talk in the adolescents about sexual health, contraception, fertility, because it's important to remember that young people with Down syndrome also grow up. They also go through adolescence. They will be meeting other people. And yes, the males may be more infertile, but actually the girls aren't. So we do need to be vigilant about menstrual disorders, how we talk to families about that, a very, very sensitive area that does need to be tackled. Mental health, we again know that children have an increased risk of ADHD and autism as well. And we need to be aware that sometimes when children are having behavioral difficulties, we don't presume it's just Down syndrome. That's the words that some people have given me. Actually, we need to assess the child properly, because then there can be different types of support that the child could reach out for. We have to start thinking about adolescents' consent and capacity. At what point can a young person consent for themselves? At what point do they have the capacity to consent for themselves? So this is a very tricky time. Transitional care is a very, very tricky period of time. And we are trying to set up more transitional care clinics. It's a bit slow. I think we're quite behind with what we should be doing. And that's often due to the way the health systems are set up. But actually the issues for adolescents are key into preparing people for adulthood. We talked about sexual health, something that we need to have tailored services for, something that we need to actually be open to talk about if we want to ensure the children aren't vulnerable. And as I said, ADHD, 6 to 8% of children have ADHD, 10 to 15% may have conduct disorders or autism. And then we get into adulthood. I mean, I've put here 10% have depression, but actually young people can have anxiety and depression as well. Children can feel excluded from their peers. They can feel lonely, and that's something that is very real. The current pandemic has impacted on children even further. They've not been able to go to school. They've been at home just sitting around, not engaging. They can't engage with the digital technology like you and I are engaging with. There's been a special system to support them in that. And so they become withdrawn. They can become very sad. They can have low mood, and this can have an effect on their appetite, which then again has a knock-on effect. And you'll have heard a lot about adulthood. I know one of my colleagues will have talked to you about this as well, about the symptoms and the health problems in adults: hearing, thyroid, autoimmune disorder, mental health problems, fertility, dementia. And there is some discussion, and this is the data that we've been trying to look at, is some discussion about a decrease in the incidence of some solid tumours and actually an improvement in the heart disease, and actually a decrease in cardiovascular disease as well. So we've been trying to tease out some of that data that's giving us some benefits as well as some problems as well. So I don't know if you can see the slide properly, but this is just to actually outline that we have to, as well as thinking about morbidity and supporting and improving health outcomes, we have to be realistic, and we have to consider what are the top causes of a person with Down syndrome passing away. Because if we know that, we can then be aware of how we can tackle those problems early. So heart defects is a key one, and then it's something that's most commonly considered people seek help for. Pneumonia, respiratory problems, that's what I've mentioned. Coronary heart disease, cerebrovascular accidents, usually when they're older, cardiac, renal problems, cancers we've talked about. So what do we do in the UK? And I know other countries, including India, also have a similar surveillance system, but maybe the implementation across the board, across the globe, is not very consistent. And we were doing some work previously where we were trying to develop a surveillance system that at least provided a minimum surveillance and then layered it so that if you had more finances, or if you had a country that could have better, more affordable healthcare, then it may be other investigations you could layer on top of it. We haven't completed that, or we haven't managed to complete it yet, but it's something I'd urge all of you to think of. For a surveillance system, is hugely important. And I've just presented all the comorbidities that a child can have. So how can we put that in practice? How do we put all the research we're all doing into practice? And that's through a surveillance system. That means from birth till death, the child or the adult is reviewed regularly, and that becomes an annual part of their healthcare system. From birth, they will have a number of different activities, such as an echo, full blood count, vision, hearing, and lots of other things. And then they'll be initially every year, and then every 2 years, they will be actually reviewed, relating what our health surveillance does with what we've seen from the comorbidities I've described earlier. In England now, what they also started doing was the pediatrician would see the child up to age 14, and then the government brought in an annual check for all individuals with learning disability. The reason again for that was because through the data analysis, we could see, and as I've presented, that adolescents and adults have comorbidities that must be picked up as well. So what we found was after the children were 14, some of them didn't come back to see us. They actually fell off the radar, nobody would see them. So by bringing this new assessment that the government have brought in, what we hope is that the primary care doctors will do an annual check on these children as they become adults as well, so we don't miss the comorbidities that I'm talking about. The challenge is that this is not routinely implemented, even though it's been recommended by the government. And we have been, again, doing some review of the services provided, and it's variable, still very variable, but it comes back to more social science research, more implementation science research that's needed. You'll see here, one of the reasons for doing that, some more statistics, is hospital admissions are higher for children with Down syndrome, mean length of stay, which means they miss out on school, they're in hospital, it's harder for the parents, and higher admissions to intensive care. This is old data from 2005, nothing has changed. It's still very, very similar. So we also did some research which was looking at if we need to develop guidelines and health surveillance reviews for children and protocols for children, where do we get the evidence for that? You know, people will challenge us and say, well, you know, we haven't got the money to look after children, parents can't afford it. You need to produce the research. And this then comes back to the responsibility of the funders as well. This research that we were doing was really to map out how many publications were carried out in a year in Down syndrome. You look at this and you think, wow, the number of publications has gone up quite a lot, quite dramatically. We should be very, very proud of this. That means we're doing lots more research. The problem is, or not a problem for everybody, but from our perspective, one of the problems is that the research is focusing particularly on prenatal publications. Huge number of publications on early diagnosis, genetics, early detection for Down syndrome. And that's where actually most of the money is going to. So it's not going on how do we improve the lives of people? What interventions do we bring in? Are people actually adhering to the recommendations that we have been developing? The research still says with the antenatal diagnosis or prenatal publications, and that was a little bit different for us actually. Again, what gets the news, what people grab in the news, what are the genetic advances in Down syndrome? Not how do we make people's lives better? I'm not saying there should be one or the other, but a balance is required. So if you look at this is actually, so it looked like the number of publications were going up for children with Down syndrome, but actually the number of publications versus other publications not about Down syndrome has gone down. So overall, what that means is that our research, and this is something for the society to consider very carefully, is overall this means that the research is actually being squeezed. We are not getting as much funding for research. The number of publications are going down. The amount of research is going down. If we do not do more research, we cannot provide the evidence to improve quality of care for the children and adults. Again, this is just a number of pediatric Down syndrome publications. As you can see, there was an increase between 2012 and 2014, and then again now a drop in the number of publications. So as I was saying, in summary, research improves mortality and morbidity. We've had a percentage decline in Down syndrome publications and a shift in focus away from childhood and over or underrepresenting of healthcare conditions. So you can just see the graph that I put at the bottom. A lot of prenatal studies on looking at cells, genetics, antenatal diagnosis, prenatal diagnosis, but much less in the world that I particularly live in. The other problem that we found was the majority of studies used a cross-sectional methodology. There were very, very few interventional studies. Qualitative studies were few, mixed method studies were few, again, because it doesn't attract funding. Most studies were concerned about the development and cognition, or about oncology or about neurology. So little about growth. And we talked, and as you can see, one of my passions is nutrition and how nutrition affects growth and the brain development as well. Little about that. Not much about the impact on mortality for children, and very, very little on child protection. Child protection is always a very difficult area to discuss, but we know that children with disabilities are more vulnerable. We know that it's challenging to look after them. We know they're vulnerable in society in general. But again, it's one of those very difficult areas to research, and it was very clear from the research that we were doing, the mapping we did, that very, very few papers were tackling child protection issues for these children. Majority focused on prognosis or looking at the natural history of Down syndrome and treatment. Fewer studies considered some of the ethical dilemmas that I've talked about, very few on economic analysis. We did one study looking at hearing aids, because you may know that there is a traditional hearing aids and there's the Baha hearing aids that go across the forehead. And even in the UK, there's a postcode lottery about who would get which type of hearing aid. And we were trying to find out why that was. Was it due to cost? Was it due to availability? So you can be in one street, you can get a hearing aid, a Baha one, and then in the next street you don't. So again, inequalities of access to care. Very little research done in that field. So current practices regarding health surveillance of children with Down syndrome. We did another study where we looked at the guidelines and protocols around Down syndrome itself and what protocols exist. And again, this was a health service type research study. What we wanted to do was say, well, surely if we know what the comorbidities are that everybody thinks exist, we seem to have a bit of consensus on that. Surely across the country, we would have one health surveillance guidance document that we all follow. And surely if we audited that, everybody across the country would be following the same thing. Unfortunately, it's not true. And we actually went and asked everybody to send us their protocols, despite there being the Royal College of Paediatrics recommendations, despite there being a Down syndrome medical interest group recommendations, which actually are not exactly the same either, and the government Department of Health recommendations for people with learning disabilities. Those are not totally identical in themselves within one country. And then if you take that a step further, you see variation in the country itself. It's not a huge variation, but it is a variation. So as we've said, there are few interventional studies, and I think we need to be thinking as the T21 is where can we profile the research we have done? Can we consider where we should apply for funding? Can we consider where the gaps are? And the families are key to that. What research do they need? Use a more participatory approach with families. So just here is a little bit. I mentioned that we were doing some research about respiratory infections. And again, there was no studies previously on focusing on hospitalization of children with respiratory infections. There was a lack of understanding of the burden of respiratory infections on children with Down syndrome. And that's why we took this research forward. Again, it was a data analysis type research project. Ideally, what we want to do is prospectively follow children up. But again, funding for that is very limited. The key findings were that there was limited evidence on interventions to prevent or treat respiratory infections. This was work done by my PhD student. Children with Down syndrome have increased respiratory tract infections and healthcare utilization. The children are prescribed more antibiotics in primary care. They have a higher risk of hospitalization. And there's some evidence that if we prescribed antibiotics for children in primary care for the very young, the 0 to 1 year old, that we might prevent some of those hospitalizations. This wasn't so true for the older children. And we don't want to promote over-prescribing either, because we really don't want to have the antimicrobial resistance scenario, which could happen if we actually did not consider the appropriateness of the antibiotics. So we're not advocating that everybody should get antibiotics. That is not what we're advocating at all. I'll follow through with that. So this is just to show you the pediatric service specification, which the Royal College of Pediatrics brought out. So this was actually a service specification for, so it wasn't a guideline for surveillance as such, it sort of includes that, but it was what should the services be doing, how should they be set up? And this is some, maybe many of you may want to look at if you're setting up your own services, to consider what are some key recommendations we thought were important. This is the step-by-step guide for primary care doctors in the UK, the annual health checks. This is not just for Down syndrome, but also for many of the other individuals as well. But like I said, there's not, if you see this chart, which you can't see in detail, but I can just give a summary, but basically what it's saying is we have all these different guidelines. There's commonality in a lot of the core aspects, but actually there's differences in some of the areas, and that is not helpful. It's actually very, very confusing. And we've been advocating NICE in England, the National Institute of Healthcare and Excellence, to have one guideline for the whole country. We haven't managed to do that yet. We've also been trying to get, I've been Transport Down Syndrome International with their work of trying to get an international guideline as well, and that's in progress, but slowly, very slowly moving forward. So why do we need that? It's hard for parents to self-report what's going on in their families, differences between recommendations, guidelines are incomplete, and we really don't know what clinicians are therefore following or not. Yes, some of the specialists are following similar guidelines, but not all children see specialists. So some of the findings in that were, particularly in the area of cardiac assessments, there was a difference in cardiac assessments. Some were, adolescents were getting cardiac assessments, some were not. Sleep disordered breathing, again, a difference. Some were having their children assessed for sleep disordered breathing. Some of the guidelines didn't routinely do that. Transition, some of the pathways mentioned it, some didn't mention it. Those were the key areas of focus. So in the end, because I know I'm running out of time, but in the end, what I'm trying to bring to your attention is it's a complex story. In summary, we need to be looking at children as a life course event. We need to be very, very on our toes to pick up the comorbidities. We need science to provide us the basic science information, so we know what the comorbidities are, we know how to treat them, and we need the implementation science as well, and we need the social science research as well, and we need to bring that whole picture together. It's not just about health, it's about health, education, social care, an integrated approach to looking at a child. We need the parents to be at the center of this, participatory research, parents driving the agenda, telling us what their needs are, setting the agenda for research. And if we do that, I know that we could tackle some of the gaps that still exist in the research that we do. Thank you very much.
Thank you, Professor Lakhanpal, for this beautiful and excellent deliberation. And I think this deliberation will enrich particularly our parents, caregiver, and the relative family member, as well as all the researchers across the world. And all the participants are actually putting a lot of questions in the chat box, but I know that we have stringency of time, and Professor Lakhanpal told me that she has another meeting to join, and we have our task force, COVID-19 task force meeting within a couple of hours by T21RS. So I have to let her to go, but still
I've got a little bit of time, about 5 minutes.
Yeah, 5 minutes is enough for asking 1 and 2 questions. Picking, well, the first question is, there is any correlation between the Down syndrome and occurrence of T1 and T2, type 1 and type 2 diabetes?
Yeah, so diabetes has been shown to be increased in children with Down syndrome, particularly as they go through adolescence, and that's what some of our data is trying to tease out. The problem is that we don't, I think across the world, we need to have a better system at collecting data on individuals with Down syndrome so that we can actually analyze the data in more detail. We are at the moment relying on databases that we're using secondary data from what we've got, and we're trying to pull that data and get as much information. But yes, there is a correlation with diabetes, and that's why it's so important, because from a South Asian perspective, we know that anyway, South Asians have a higher risk of diabetes. On top of that, if like I explained, the obesity, if that comes in as well into the agenda as well, that increases it further. And then the Down syndrome will increase it any further. So yeah, we need to be very, very careful about early recognition and identifying individuals early about that, but also trying to prevent the diabetes as well.
Well, another question is there any system biology approach to better understand the DS and its associated comorbidities?
The system biology approach, yeah, because we're doing mainly data analysis. I have to say, the data analysis is the starting point, because actually, unless we know what the comorbidities are from a data perspective, we actually can't go on to the next step. So I think once we are, I'm not a basic scientist, so it's a little bit more tricky for me to answer that question. The way I usually start is, what does the data tell us? Does they tell us that there is any increase or not? Find out what their comorbidities are. Let's then work with the scientists to find why that may be. There will be, as I heard your speaker talking before, is it due to nutrition? Is it due to folate? Is it due to X, Y, Z? What is the reasoning behind it? How does the genetics impact on these comorbidities? Is that what's going on? So there will be a lot of research that needs to be done to find out why. But first, the first step is what we're trying to do is find out what those key comorbidities are and then move to the next step.
Well, and the last question is, what is your take home, what is your idea about formulating government policy on bearing women of certain age for conceiving?
Sorry, could you just say that again?
Yeah. Is there any fixed government policy for having a children or conceiving so that we could avoid Down syndrome childbirth?
So this comes back down to a very ethical question, is that I never talk about avoiding children being born with Down syndrome. Children with Down syndrome are beautiful children, and we value every child who comes into the world. But everything's a choice, and everything's a choice for parents. And so I think that what we do is we present the data to parents, we present the science to parents, we tell them what the age is, where there are higher risks to parents, and then parents will make a choice of what risks they are able to take or prepared to take. Everybody's circumstance is different and everybody's lives are different. And then once they know the risks, we do now know that there are tests that can be done to identify if individuals are carrying a child with Down syndrome. But these are very, very complex personal choices. And I would personally never want to take the freedom of choice from mothers, families, or anybody else in that way. And so I would not leave it to the government to do that sort of thing.
All right. Thank you, Monica. That's a nice presentations, and there are a lot of questions, but still, I hope next time when Monica will come to India, we'll arrange one seminar at University of Calcutta, so that you people, those who are really interested, could meet her in persons and could satisfy your query and quest regarding Down syndrome and your children. And if you have any further questions, please send me in mail and I will forward those mail to Monica so that you people get your answer. So thank you, Monica, once again.
Thank you very much.
Thank you. We are grateful and see you in the COVID-19 meeting. Thank you.
Bye-bye.
Bye-bye. So participants, this is the end of our sessions. Thank you for being with us, keeping your patience for the long time and hearing those lectures. So if you will with us, then once again, we'll design such kind of the webinar, taking experts from the different parts of the world to deliver the latest and updates in the Down syndrome research and different health issues. And that will help our society, our parents, our caregiver, and obviously to young researchers to boost up, to take the Down syndrome as a subject in their future research career. Thank you for being with us. Please put your feedback in your given feedback form in the chat box, and you will have your participation certificate by 15th August. We need 2 weeks to work, and then we will send you all the participation certificate either by mail or putting them in the common Telegram group. Whatever, you will get your participation certificate. Goodbye, thank you, and congratulations. Thank you. Have a nice weekend.